Today's lecture is going to be all about bacteria. So, I have seen many of my friends and colleagues who have been preparing for the NBDE Part 1 and they're having a difficult time with the microbiology portion. So, I thought of making a video of the microbiology most important topics and most important bacteria, and I've tried to make it as small and as short as possible, just putting the most important stuff only. So let's begin. And this is going to be a two-part video. So this is the first part of the video. Okay. So before we go on with the bacteria, let us just know a little bit uh, difference between the eukaryotes and the prokaryotes. So the prokaryotic cells are the primitive cells, and most of the bacteria fall in this group, and the eukaryotic cells are the more advanced cells, the human body cells and the animal cells or the plant cells or maybe the fungi and all of them. They're a bit more, I would say, more complex than the prokaryotic cells. So the basic differences are these, and there might be a few questions asked about this. So it's better if we just go through the differences really quickly. First, the nucleus. The prokaryotic cells do not have a nucleus. The eukaryotic cells have them, and the nucleus is membrane bound, but the prokaryotic cells do not have a nucleus. All the genetic material of the prokaryotic cells is present in the cytoplasm. The DNA found is present in the cytoplasm and not in the nucleus, where we, like in the eukaryotic cells, it's found in the nucleus. That's what the nucleus is, like the brain of the cell. It stores all the important data. So the prokaryotic cells have their DNA that is circular and naked. That naked means it's not bound to proteins. So that is found in the cytoplasm. It's floating in the cytoplasm, and it's sometimes it's called as a nucleoid. And the eukaryotic cells have their DNA in a linear form, as we see it, what actually we've studied about the structure of the DNA. And that is bound to proteins. Those are known as histones. And they're found mainly in the nucleus. So the nucleolus is absent in prokaryotic cells, whereas it is present in the eukaryotic cells. The prokaryotic cells are they're gonna lack most of the organelles like the mitochondria, the nucleus, the ribosomes, and everything. It's a primitive type of cell. If you compare like the machines of today and the machines of the old age, it's something like that. It's really stupid comparison, but it's something like that. Then the DNA replication is gonna happen in the cytoplasm because the DNA is present in the cytoplasm. It doesn't have any room to get into to reproduce, if you know what I mean. Does not have anything. It has to do it in open. It has to do it in the cytoplasm. And whereas the eukaryotic cells, they have a place where they can do the DNA replication, that's inside the nucleus, because they're already present in the nucleus. They're not going to go out of the nucleus into the cytoplasm just to replicate. They're going to replicate in the place where they are already. Then the reproduction occurs by binary fission, and in eukaryotic cells it occurs by mitosis or meiosis. So this one is really important. You can find this in some questions like prokaryotic cells reproduce by which of the following method. So it's going to be binary fission. I think even these can be a potential question. DNA replication occurs, or where is the DNA found in prokaryotic cells, or what is the genetic material of the prokaryotic cells called? So it's quite a simple thing. Just if you memorize this table, I think you're good to go. Then one of the major differences of cell wall are the prokaryotic cells are made up of peptidoglycan. It's a complex polysaccharide with protein components in it. So that is not present in the eukaryotic cells. And if it is present, if the cell wall, if like cell wall is not present in most of them, the cells in our body don't have a cell wall. It's like the animal cells don't have cell wall. If they have a cell wall, mostly they're found in the fungi. So they would contain chitin. Then organelles, as we said, there are no organelles found, like no specific organelles found, like those are found in the eukaryotic cells. And in the eukaryotic cells, if they are found, most of them or some of them are membrane bound. Um, Size, as we said, it's a primitive cell, so just imagine it's old age and it's really small. It doesn't have much function, it's a smaller type of the cell, and the eukaryotic cells are usually larger because they've got all those big heavy organelles and they've got all those parts which do the work for them. So that's the reason they're larger. And they're usually haploid, that is, they have a single gene, and eukaryotic cells are diploid. So that's not much of a deal, but yeah. Okay, this is a simple diagram. As you can see, the prokaryotic cell, it has the nucleoid. That's the genetic material. It's right in the center. Does not have a nucleus. Does not have a nucleolus. Doesn't have any other organelle. It's all floating in the cytoplasm. And whereas look at the eukaryotic cell, it's much more complex. This is the endoplasmic reticulum. This is the mitochondria, and various other lysosomes and everything. So it has a well organized structure. Okay, then we go on to a difference between the gram positive and the gram negative bacteria. So there are basically two main types of bacteria, the gram positive ones and the gram negative ones. It's upon those who take the gram stain, they're the gram positive bacteria, and those who don't take the gram stain, they're the gram negative bacteria. We don't need to go much into detail about gram staining, but so the major difference between gram positive and gram negative is that the gram positive organisms, the cell wall, it contains teichoic acids, or and it is at a thick peptidoglycan layer. You can, and this is of importance here. The peptidoglycan layer is also called as the Muirian layer. So if you find this in a question, don't get confused. It's gonna be the peptidoglycan layer. So, and this can sometimes be asked, can be sometimes be tested on, like what is the layer made up of. So they're made up of alternating units of N-acetyl glucosamine and N-acetyl muramic acid. So I think you probably should remember these two names, N-acetyl glucosamine and N-acetyl muramic acid. The gram negative one is a little more complex than the gram positive. It has a thinner layer of it has a thinner layer of peptidoglycan than the gram positive. Over the peptidoglycan layer, it has a lipopolysaccharide layer which is not present in the gram positive bacteria. It's only present in the gram negative bacteria. So this is the layer which is responsible for making the endotoxins. It's the layer which sheds off and forms the endotoxins. So just remember, lipopolysaccharides are present in gram negative. So gram negative bacteria are the ones which produce endotoxins. There are some gram positive which produce endotoxins, but if you get a question on the test where regarding this, it's going to be gram negative bacteria which produce endotoxins. This is a diagram for it, and this is a reference from where I got the diagram. So this is a typical gram-positive bacteria. It's got a thinner layer of it's got a layer of peptidoglycan than the gram-negative bacteria, and as you can see, there's teichoic acid, which is not present in the gram-negative bacteria. The gram-negative ones have got the lipopolysaccharide layer, which the gram-positive ones don't have. So all you got to remember between these two is the lipopolysaccharide layer is present in the gram-negative, whereas the teichoic acids are present in the gram-positive, because these are the areas which are tested on a lot. Okay, we go on with the gram-positive bacteria, and that first one is the gram-positive cocci. So cocci or coccus are it, they are circular organisms, and they can be named after their colonies. If they're present in a grape-like cluster, it's called a Staphylococcus, and if they're present in chains, they're called Streptococcus. So these are the two main gram-positive cocci. We go with Staphylococcus first. The most common one is the Staph aureus. That's the most common pathogenic organism. most commonly found on the skin. This can be a question. It's a important question. It can be asked, like, where is Staphylococcus most commonly found? So it's gonna be skin, because you can remember it this way. When we go in the further slide, it causes various skin diseases, so you can remember it that way, that it causes skin disease, so it has to be present on the skin. So as I talked earlier, its colonies are like a grape-like clusters, so that is the reason it's called Staphylococcus. They cause suppurative infections, that is, those infections which produce a lot of pus, so of the skin, the joints, and the bones. So you can get a question on the test, which would be like, which is the most common causative agent of osteomyelitis? So the answer is going to be Staph aureus. That is the organism which causes osteomyelitis. Now, it can be coagulase positive or negative. You know what? Okay, let me explain in short about coagulase. It is an enzyme produced by the Staphylococcus. So there are various types of Staphylococcus. There's Staphylococcus aureus, Staphylococcus epidermidis, Staphylococcus saprophyticus. So these organisms, they can be coagulase positive, which means that I'm sorry. I think I there was some problem. Yeah. So they can be coagulase positive or coagulase negative. That means they produce the enzyme coagulase, or they don't produce the enzyme coagulase. So Staphylococcus aureus is coagulase positive, whereas the epidermidis and saprophyticus are coagulase negative. You don't need to remember this. If you it's okay if you can't remember it. And they're catalase positive and organisms. They even produce catalase. So catalase is the enzyme which breaks down the H2O2. That's a free radical produced by organisms. So it is actually toxic to the organism. So those organisms which have catalase enzyme, they can degrade H2O2, and they can protect themselves. So Staphylococcus is catalase positive. That means it produces catalase enzyme, and that one produces. You can get a question like, which one of the following is catalase positive, or you can get a question like, which one is sensitive to H2O2, or which one is affected by H2O2. So the this one is catalase positive, and Streptococcus, which we study in the next slides, is going to be catalase negative. So the pathogenic features, they produce protein A. It's present in the cell wall of it. It binds to IgG antibodies. Don't need to remember much. Just remember Staphylococcus produces protein A. It cannot attack intact skin. There has to be some trauma or laceration or something to for the bacteria to get inside the body, and then it's gonna attack the body. It cannot. If you if you have a bunch of staphylococcus in your hand, and if you're gonna rub it on your skin, if you don't have any laceration or anything, it's not gonna enter your skin. The skin is one of the best dressing. So that's what given in one of the surgery books. Don't need to get into detail of that, but yeah, it cannot attack intact skin. So once inside the body, it secretes enzymes and toxins, and it's gonna infect the host. It's gonna cause the disease. Then it produces another enzyme called beta lactamase. So this one is really important, because the organisms which produce beta lactamase, they're not gonna be affected by the penicillins. They're gonna, you know, cause a reaction, and they're gonna degrade the penicillin. They're not gonna be affected by penicillin. So, just remember, these organisms are beta lactamase producing and organisms. So the diseases, they produce boils, carbuncles, and abscesses. So boils are big infected pimple like things. Then it causes a fatal disorder called Staphylococcus scalded skin syndrome. That's SSSS. And it produces another fatal disease called the TSS. That's a toxic shock syndrome. Then as we talked earlier, it causes osteomyelitis. Is the most common organism which causes osteomyelitis. It also causes bacterial endocarditis and staphylococcus food poisoning. It's an acute onset food poisoning. Now we have the other staphs. They are not much tested on, but in a rare chance you can get a question on these. Like the epidermides, staph epidermides, is associated with the infections, which are mostly associated with medical devices like, the prosthetic valves or those kind of medical devices. They're present on them, and they can cause nosocomial infections. And then we have the saprophyticus, the Staphylococcus saprophyticus. It produces urinary tract infections. These two staphs are coagulase negative. So, coagulase test is used to differentiate between different types of staphs, and catalase test is used to differentiate whether the organism is Staphylococcus or Streptococcus, because Streptococcus is going to be catalase negative and Staphylococcus is catalase positive. Then we have the Streptococcus. It grows in chains or pairs. Strepto meaning chains. So it's the most numerous organism of the oral cavity. It's the one which causes dental caries. So you're gonna see a lot of questions on these because we are dentists and we're gonna give the test for, a dental test. So you're gonna see many questions on these. So it grows on glucose medium and produces lactic acid. So that is one of the reasons this is how caries is produced. You eat something, it gets degraded into glucose. You eat a lot of candies and sugar and stuff. The streptococcus present in the mouth, it's gonna it's gonna start acting on that. It's gonna start producing lactic acid, and that is the thing. The lactic acid starts degrading the tooth. That is how caries are formed. So um, you can sometimes get a question on which of the following organism, when put in a glucose or sugar solution or medium, drops the pH. The solution drops the pH and it becomes acidic. Then the answer is going to be Streptococcus because it produces lactic acid from glucose. It has got an enzyme called glucosyl transferase. We're going to see it in the next slide. Okay, they're classified based on the action of blood agar. Blood agar is a medium. You put those streptococcus in it, and then on the type of hemolysis they produce on the agar, they're going to be classified into alpha hemolytic, beta hemolytic, and gamma hemolytic. We look at the alpha guys here. So a mnemonic to remember what type of organism does what type of hemolysis. You go with A, alpha hemolytic. It does almost, just remember the word almost. That is, it does a partial hemolysis. It does not do a complete hemolysis on blood agar. It does a partial hemolysis. That's And then we have the beta hemolytic organisms. They do the best. Just remember the word best. That is, they do complete hemolysis. And gamma hemolytic, that is, it's just G for garbage. So just remember, doesn't do any type of hemolysis on blood agar. We have the alpha guys. They produce a green color on the blood agar. An easy thing to remember if you can. Like one of the most common organism is the strep viridans and the strep pneumoniae. So if we got the strep viridans, think verde is green in Spanish. I've been learning a bit of Spanish in the past couple of months, so verde is green in Spanish. So just remember viridans produces green color on blood agar. We got a strep pneumoniae. They primarily live in the nasopharynx. That can be asked sometimes. Strep pneumoniae colonizes in which of the areas, and that's going to be nasopharynx. So, various types of strep viridans. There are mutans, mites, salivarius, sanguis. These, most of them are associated with caries. Gotta remember them. They're normal flora of the oral cavity. Salivarius is found on the tongue. This is asked sometimes, so better remember it. Streptococcus salivarius is found on the tongue. Mutans is the ones which causes caries. That is, it is acidogenic, which means it produces acid. We talked in the previous slide. You get glucose, it's gonna eat up those glucose and throw up some lactic acid, and that's how caries are formed. And it's got the enzyme glucosyl transferase. And I'll put it in red because this is really important. Every now and then you'll get a question on this, that which of the following organism produces glucosyl transferase, or you can get a question, Streptococcus produces caries with the help of which of the following enzymes, and the answer is going to be glucosyl transferase. Then viridans streptococcus can cause SAB, that's subacute bacterial endocarditis. Now this one is a tricky one because you can imagine this organism is present in the mouth, and how is it going to affect the heart? So yeah, you gotta remember it, because if you get a traumatic extraction or something, these organisms, they're gonna enter the bloodstream, and it can cause subacute bacterial endocarditis in those persons whose heart valves are already weakened, or they have already had a prosthetic valve or something like that. I don't need to get into much detail of how that happens, but just remember Viridian streptococcus can cause subacute bacterial endocarditis. Really important for dentists, because you got to give prophylactic antibiotics to those patients. So um, now we have the beta guys. They do complete hemolysis on the blood agar. It includes Streptococcus pyogenes. That is the most important streptococcal pathogen. um how do you differentiate it from other strep? You got an infection, you scrape out a tissue, and you send it to the lab, and then you want to know what it is. Then what the lab is going to do is they're going to grow it in some kind of medium, and they're going to, if when you want to differentiate it, they're going to put the antibiotic bacitracin. So you put it inside the medium, and all the other streps, sorry, they're gonna live, and this one is the streptococcus pyogenes is gonna die because of the bacitracin. So that's how you can differentiate from other streps. The diseases caused by it are pharyngitis, another important one to remember, otitis media, peritonsillar abscess, scarlet fever, glomerulonephritis, and rheumatic fever. Now, you can get a question asking about what is the sequelae of streptococcal pharyngitis, because that's how the answer is going to be rheumatic fever. So that's how rheumatic fever happens. Earlier in your life, a person gets strep throat or streptococcal pharyngitis, and he's susceptible to get rheumatic fever in the future. So that's the sequelae. You can get that question too. You have the gamma, that's garbage. Just remember G, gamma, garbage. No hemolysis on. And we have the gram-negative cocci. Two important gram-negative cocci are Neisseria meningitidis and Neisseria gonorrhea. As you can see from the name, Neisseria meningitidis causes meningitis, and Neisseria gonorrhea causes gonorrhea. We got the N meningitidis. It's a diplococci. Diplococci is typically the two cocci present together, so they're gonna always go in it together. Both of them can't live without each other. Something like that. So you got a diplococci. They're non-motile. They're non-spore forming. It's okay if you don't remember those two, but it's just to put it in the slide. You got the virulence factors are the antiphagocytic capsule and endotoxins and IgA protease. IgA protease is the one which destroys immunoglobulin A. So it is transmitted through respiratory droplets. It can cause meningitis and meningococcemia. That's a fatal condition, septicemia and meningitis and everything, and it's a really fatal condition. It is associated with Waterhouse Friderichsen syndrome. I don't know if I pronounced it right or wrong, but you gotta remember this one because you can get questions on this. So basically, what happens in this syndrome is there's necrosis of the adrenal gland. That's a difficult condition where adrenal gland is really important for your life, and there's necrosis of the gland. So what happens is it causes hypotension. There's sepsis because all the meningitis, the organisms are going to be in the blood. They're going to cause infection of the whole body, cause hemorrhage, and it's a very fatal condition. So you gotta remember this one. Then we have the N gonorrhea. It is a beta lactamase producing organism again, so it's not going to be affected by penicillins. There's no meaning of giving this patient penicillins because it's not going to affect them. Gonorrhea is a sexually transmitted disease, so um, not much information about this one. You just got to remember this another thing, that's ophthalmia neonatorum. If a female has gonorrhea and she's pregnant and she gives birth to the child, then there's high chances that the child can get infected by, the infected birth canal, and the child is gonna have an eye infection with this. That's called ophthalmia neonatorum. It's a fun fact. Earlier, in the earlier times, they used to put the doctors used to put silver nitrate drops in the eyes of newborn kids, no matter if the mother had gonorrhea or not, just to prevent ophthalmia neonatorum. It's just a fun fact. It's okay if you don't remember that. Another important thing, it can cause septic arthritis. They can ask you a question that a young female or a young male, sexually active male or female, who's got a septic, who's got septic arthritis, and the doctor or the physician takes, aspirates from the joint space and sends the pus for culture. Then what do you find in the pus? So the answer is gonna be something like, you're gonna find N gonorrhea, or you're gonna find Diplococcus or something like that. So just remember, it can cause septic arthritis in a sexually active female or a male. Okay, so we're gonna have the gram-positive bacilli and the others in the part 2 of the video. Stay tuned for second part of the video.